all-aqueous droplet engineering for biomimicry and bioencapsulations. Compartmentalization is the key to biomimicry, cytomimicry, and also bioencapsulation. So as early as during the origin of life, the world is very simple. It just has a primordial soup that should be very dilute and very stochastic with different biopolymers and different precursors. The question is, how can these different biopolymers, precursors be concentrated and compartmentalized into such sophisticated structures that we see in the world right now? And one of the way that compartmentalization can happen is through the so-called liquid-liquid phase separation. We start with a very simple homogeneous liquid solution, and then during phase separation, different structures, different droplets, different shapes start to emerge. And this has been something reported by different researchers such as Cliff Brangwyn and Anthony Hyman, who first suggested this approach and suggested how life began using this approach. And also in cells, there are lots of observations of these intracellular liquid-liquid phase separations. For example, in pea granules, they can behave like liquids, and the fluid-like behaviors of nucleoli also suggest that there are liquid-liquid phase separations in these liquid membranous organelles. So the non-membrane bound compartments may be remnants of ancient structures that served to spatially confine and organize chemical reactions. This is one of the emerging beliefs right now. And to talk about liquid-liquid phase separations, we first have to talk about two common types of LLPS. First one is known as the associated LLPS. It's driven by specific molecular interactions, charged polymers. For example, this could be electrostatic, cation-pi interactions, dipole-dipole contacts, pi-pi stacking, etc. And this can occur at very low polymer concentrations, because if you have the molecules, if they associate with each other, even though they may be at very low concentration, you can get them to form new phases. But the problem is that with them, they may also inhibit certain biochemical reactions, because these interactions may also interfere with some of the reactions. For example, you can see in these papers that they can destabilize the oligonucleotide duplexes inside coacervates. They can also inhibit RNA polymerizations inside the coacervates. Another approach to do LLPS is so-called segregative or non-associated LLPS. Essentially, this is driven by non-specific molecular interactions of neutral polymers through mainly van der Waals interactions. The advantage is that now you can enhance biochemical processes because these don't actively participate in the chemical reactions. However, the disadvantage is that you typically have to have pretty high polymer concentrations or pretty high additive concentrations for that to happen. And so the challenge is, how can we trigger and maintain these non-associative or segregative LLPS under very simple conditions such as those in the prebiotic world. With that, in my talk, I will mainly focus on the use of segregative and non-associative LLPS, using an example called the aqueous two-phase system. Essentially, you can add polymers or salt to water, even though these polymers and salt are soluble in water, but at high enough concentrations, they can go from, for example, initially at low concentrations, they are miscible with water, so you just have one single phase, but as you increase the concentrations, the solution will phase separate into two immiscible phases. One will be rich in polymer X, the other one will be rich in the polymer Y or salt. And microscopically, you can see the droplets forming as a result of the immiscible aqueous phases. Macroscopically, you can see clear interfaces between the top and the bottom aqueous phases. And this is the so-called aqueous two-phase systems formed by a segregative LLPS. And the interfaces between these aqueous solutions are not the same as the traditional interfaces between air and water or water and oil that are immiscible, because the two phases, they are still somewhat miscible. For example, small molecules such as water molecules can still diffuse across the interface. So the interface is somewhat diffuse. So if you look at, in this case, we have dextrin and gelatin ATPS, aqueous two-phase system, you can see a gradual reduction from very high gelatin fraction to zero gelatin fraction across the interface, and the polymer concentrations may have a continuous change in the concentrations across the interface, and they may never have a sharp or step change in the concentrations of the two components. So this is a way to characterize the aqueous interfaces. So in this talk, I'll talk about how we can use aqueous two-phase system to achieve compartmentalization. In particular, I'll talk about how we can use droplets as the compartments, and how can we stabilize these droplets, how can we mimic some of the biological behavior, such as the division of cells, can we do that in droplets, and how can we, for example, compartmentalize microreaction, just like prebiotic compartmentalized droplets have to achieve. And I will start with first the droplet stabilization and divisions. So one of the challenges in working with these ATPase droplets is how to stabilize them. You can see if we make an emulsion using the two immiscible aqueous phases, by shaking them, the droplets will merge with each other and form larger and larger droplets. So in this experiment, we try to add lysozyme at different degree of polymerization. So starting from monomers to unfolded monomers, prefibrillar oligomers to mature fibrils. We use these different stabilizers to try and stabilize the aqueous emulsions. With the monomers and also the oligomers, you can see that the shaken emulsions quickly merge with each other to form larger droplets. Only in the case of the use of the nanofibrils, the droplets can remain small emulsion droplets without severe coalescence. And this is one area that we were curious as to understanding why they can provide such good stabilization. So to understand that, we look into these nanofibrils more specifically. And if we add small amount of the nanofibril plus a dye that can tag these nanofibrils, for as low as 0.025 weight% of the nanofibrils, we'll be able to stabilize the all aqueous droplets. You can see the fibrils and the dye are concentrated in the interfaces of these droplets. This suggests that all the nanofibrils that we initially add to the droplets get adsorbed to the interfaces of the droplet, and not so much, or if any, are left either in the droplet or outside the droplet. If we just add the dye but not the fibril, the dye will stay inside the droplet. So it is not because the dye prefers the interface, it's because the fibril prefers the interface of the aqueous droplets and the dye prefers the fibril. And if we look into the droplets on the surface, you can see lots of these nanofibrils crowded on the surface, providing the stabilization that we observed. And to account for this stabilization and understand how it's working, we try to vary the compositions of the ATPase by varying the concentrations of the PEG and the dextran that are used to form the ATPase. So again, at low single phase, at low concentrations, we only have a single phase. But as we increase the concentrations of PEG and dextran, we start to have two immiscible phases, and we observe the stability of the emulsion droplets at different compositions. Only within this gray area, the dextran in PEG emulsion droplets are stabilized. Outside of this gray area, they form either destabilized PEG in dextran emulsion droplets or destabilized dextran in PEG emulsion droplet. And you can see these two different boundaries. One is by this blue dotted line, the other one is by this red dotted line. If we go from the single phase region all the way outwards, if I try to remind you of what we talked about, in this single phase region, we don't have any phase separation, so there is no interface. The interfacial tension should be 0. But as we increase the concentration, we start to have interface, and the interface start to have a higher and higher tension. So if we gradually move from this 0 interfacial tension area to these blue dotted line, the surface tension was measured to be about 6 micronewton per meter. And so what this suggests is that we can only stabilize the emulsions at higher surface tension. This is not surprising because the surface tension or the interfacial tension multiplied by the total amount of interfaces give you the energy of the system. If the energy is sufficient to absorb the fibrils at the interface and they will not be knocked out or knocked away by thermal fluctuations, then the droplets can be stable. And on the red dotted line here, we also notice that if we go from dextran in PEG emulsion droplets to PEG in dextran emulsion droplets, the PEG in dextran emulsion droplets tends to be less stable. This is also because the nanofibrils actually prefer the PEG phase. So that means this is what we call partitioning. The nanofibrils partition to the PEG phase. So if the nanofibrils are stabilizing the droplets against coalescence with each other from outside the droplets, the stabilization ability is better than if the nanofibrils try to stabilize the droplets from within the droplet. So this is what we found. And we also look into whether it's possible to enhance the stability of emulsions that would otherwise be unstable. For example, we started in this area that was in the white area rather than the gray area, and then we provided conditions for the protein fibrils, initially not enough to stabilize the emulsion, but we allow them to polymerize and grow. And as a result of their polymerization and grow, suddenly some of these emulsions in this area that is now gray, these emulsions are now also stable. So this is one of the first examples that we can use protein polymerization, kind of simulating growth, to enhance the stability of the ATPase droplet. So this was one of the surprising findings we found. And so far, if you remember, we were only using very dilute fibril concentration. What if we increase the fibril concentration tremendously? For example, we go from having almost no nanofibrils inside the droplets because they all migrate to the interface, but we can also add so many nanofibrils so that even the droplets inside, they can form a network. What would happen? What we notice is that with these concentrated protein nanofibril in the droplets, the droplet exhibits surprising behavior. They protrude and eventually can divide into smaller droplets. This is contrary to what you would predict for emulsion droplets. Typically, for thermodynamic reason, they tend to coalesce and become larger rather than becoming smaller or splitting into smaller droplets. So what's happening here? This is driven by the osmotic force applied to the nanofibril network inside the droplets. As we suck water out of the droplet osmotically, we are exerting a force to the network inside the droplet. And as a result, the network, because the network inside the droplet is connected also to the nanofibril network at the interface of the droplet. So it's pulling the whole droplet and sucking the immiscible aqueous phase inside the network outwards. It's like having a sponge soaked with water, and you put the water-soaked sponge into oil and squeeze the sponge. The force that you applied to the network, to the sponge, would squeeze out the water, and the water would be immiscible with the surrounding phase, much like in this case. And we notice that you can vary the fibril concentrations, you can vary the degree of dehydration to control the degree of formation of the daughter droplets or the droplet division. And we can go from having no division to single division to multiple division simply by varying the shrinkage ratio and also the initial fibril concentration. So overall, this may have implication on understanding how cells would divide or would bud. But of course, we understand that in our system, there are no membranes, unlike in biological cells or in other cells, where the cell membrane play a very important role. And this work was done by my former PhD student, Song Yang, who is now a professor in Shanghai Jiao Tong University, and in collaborations with my collaborators from Cambridge and also from Georgia Tech, and of course, some of the funding agencies. And besides this droplet stabilization and division, another very important aspect would be the droplet reaction. So one of the challenge in using these compartmentalized droplets and explain how they could lead to such sophisticated structures in the prebiotic world is how you can have reaction. For example, how nucleic acids such as DNA and RNA can amplify. And so people have been looking into how we can do a biotic synthesis in these compartmentalized droplets. For example, how you can synthesize peptide, how you can synthesize RNA nucleotide, how you can synthesize or condense peptide by going through wet-dry cycles, because wet-dry cycles presumably is very simple. You're only changing the temperature, just like when the sun goes up and goes down and you have sunset and sunrise, and that may lead to different temperature cycles, and that may be something that you can also have during the origin of life. And we have done a work to study this by putting the components in a droplet, and then by using evaporation, we try to assist the segregated phase separations and try to mimic the prebiotic compartmentalizations. So let me go into detail about this. So essentially, if you have a droplet with components that can facilitate the aqueous phase separation or the segregative LLPS that I mentioned, so you can have two different regimes. For example, in regime 1, you can see that you form lots of small droplets around the rim, and then these droplets can merge with each other, and eventually, as they move inwards to the core of the droplets, they can merge all together, forming this core shell looking like droplet at the end. This is all driven by the segregative LLPS. Another case is that you can trigger, as water evaporates from the droplets, formation of tiny dotted droplets from the rim, but they don't merge with each other quite as much. Instead, they shed a lot of small droplets into the core, and as they go into the core, eventually they become bigger and they coalesce with each other, forming in the end also a core shell drop using a different route. Which route you can take will depend on the relative concentrations of the PEG and dextrin that you have in the starting composition of the droplet. If you have something like in here, in the middle between PEG and dextrin, then you can go through regime 1. But if you, for example, have a higher PEG concentration and lower dextrin concentration, then you can form in this regime 2. In this case, the red part is the rhodamine B labeled PEG, and the green part is the FITC dextrin. You can characterize the behavior between regime 1 and regime 2 in this phase diagram. And you can understand, for example, starting from the center, as you extract water from the droplet, you increase in concentration, causing phase separation, and as a result, you can have two immiscible phase. One will be rich in PEG, the other one will be rich in dextrin in F1, F2, and if you repeat that further, you can have one more step of phase separations with concentrations S1 and S2, and this is in regime 1. In regime 2, you start somewhere in here with the high PEG concentrations, and you can get to different steps of phase separation as well. And this is an approach that can be used to encapsulate DNA and RNA, as you can see here. In the top video, we also have DNA in the environment, because when you have two immiscible aqueous phases forming, one of the aqueous phases will prefer or will have a higher solubility for the DNA. So the DNA in the environment will get concentrated into that particular aqueous phase that prefers DNA. And so you can see the denser fluorescence in these droplets. And likewise, if you have you have one of the two immiscible aqueous phases where the RNA would have a higher solubility, so the RNA would partition to this particular immiscible phase, and essentially the phase separation can help to concentrate the DNA and RNA in the system, or can help to encapsulate the DNA and RNA in the system without applying external forces. You can also use this approach to achieve in vitro transcription. So here we have some DNA templates that are coded for the broccoli aptamer that can bind to this DFHBI. And you can see that because a fluorescent complex of broccoli and DFHBI will be formed upon transcription. So you can see that the transcription is happening because the fluorescence intensities in these droplets are increasing. So you can do this in vitro transcription in the droplets. Another thing that you can also do is the ribozyme cleavage. So here we use fluorophore labeled hammerhead substrate and it can be cut into 2 smaller pieces by the hammerhead ribozyme. And once you've done that, you will see green fluorescence. If they are not cut into 2 smaller pieces, they will not show green fluorescence because their fluorescence will be quenched by the substrate. So here you can see that as we play the video, initially you don't have very sharp fluorescence green, but as the reactions start to happen, you can see some of these droplets become more intense in the green fluorescence. And this is an example highlighting the cleavage of the hammerhead ribozyme. And this is a very important step because in understanding the origin of life, how RNA or DNA can be amplified in the prebiotic world, one requirement is that the RNA has to be able to be catalyzed by themselves rather than other enzymes or proteins, because during that stage in the world, those proteins or enzymes do not exist yet because they are too complicated. So this is one example highlighting that without adding any enzymes or proteins, if the concentrations of some of these RNAs is high enough, it is possible to catalyze their reactions. So hopefully with this work, we demonstrate that it's possible or prebiotically plausible to use non-associated phase separations within an evaporating or aqueous sessile droplet. And you can see that the kinetic pathway of phase separations can be observed. And this is assisted by Marangoni flow-driven hydrodynamics inside a sessile droplet that I did not go into. If you're interested, you're welcome to read our papers here. And the drying droplet also provide a model system to demonstrate the localization and storage of nucleic acid, in vitro transcription, as well as enhancement of the ribozyme activities within the droplets that we use. And this work was mainly done by my PhD student, Wo Wei, in collaboration with some former members and as well as collaborators. And this work is funded by Hong Kong U, RGC, the ITC of Hong Kong, Croucher Foundations, and NSFC. And our works also include different aspects in our group. For example, besides some of these cytomimicry work, we also do some all-aqueous multiphase system for material synthesis. We also do some microfluidics to study different droplets, manipulate droplets, and use them for single-cell studies, use them for genomic studies and analysis. And we also use our microfluidic techniques to try and mimic physiological conditions, to study cancer metastasis, to study the eye, for example, how we can use our microfluidic chips as eye on a chip to understand some of the physiological phenomena, etc. So if you're interested in this or any of the works I've just presented or discussed on this slide, please feel free to contact me at this email or visit our group webpage. Thank you very much for your time.